So I'll tell a brief overview of our work that recently published in Cell Reports. So in this work, what we tried to understand is how the SARS-CoV-2 coronavirus actually enters into the living cell, and the mechanism, like how the spike protein of this virus actually facilitate the entry process of the viruses. So, a lot of thing was known, which is in terms of the receptor that this virus actually utilize to engage first to gain entry, but the actual mechanism for entry was not known. So, in terms of the receptor, what was known from previous publications from other groups is like ACE2, TMPRSS2, NRP1. So, these are the protein molecules which were helping these viruses for gaining entry inside the cell, and these receptor molecules are present actually expressed in lung epithelial cell lines. So, that is why in lung mostly these viruses were heavily populated in comparison to other cell in our body. So, what we were trying to understand is once this virus actually engage with the cellular receptor proteins, and how the interactions of this receptor protein with the spike protein of the virus actually helps the virus to enter inside the cell. And so, the entry process happens by a steps which is called membrane fusion, and in this process the virus membrane of the SARS-CoV-2 actually fuses with the cellular membrane. And the idea was that the membrane fusion can happen either at the plasma membrane or at the late endosome phase, where the cellular endosomal membrane will fuse with the viral membrane, and then after the fusion the genetic material of the virus will go inside to the cell for making new viruses. So, what we find that for this membrane fusion to take place, low pH is highly required. So, we made a in vitro assay to understand the effect of each receptor proteins, and we find that the low pH is very important for membrane fusion process, and that indicates that the fusion process actually happening mostly in the lysosomal stage. So, virus actually utilize the lysosome for getting entry, and apart from low pH, the other things that we find which is very important is calcium. So, the role of calcium is very, very important. We find that the calcium ions actually helps this membrane fusion process, and we find that the calcium, if we somehow block the calcium inside the lysosome using some small molecules, and that actually affects the viral entry inside the cell, and viral couldn't enter if we use some small molecule which actually blocks the calcium ion channels in the lysosome, and the virus entry process is inhibited. And we also use some single molecule techniques, structural biological approaches using FRET and anisotropic measurement to see literally how the spike protein actually changes the conformation from pre-fusion state to the post-fusion state. So, pre-fusion state is the before fusion state, and post-fusion is after fusion state. So, altogether we find that low pH and calcium is the ultimate regulator of the SARS-CoV-2 viral entry. So, in future what we are trying to understand how these things is actually related for the new incoming variants, like for Omicrons and sublineage new variants that which are coming, and we are now trying to understand how the role of pH and calcium is actually affecting their entry processes. So, thank you.